Ready to go?
Yeah, we're recording. So everybody.
Great. Well, thanks for having us. This is certainly a first time for us. We do these virtual tours every year, or not virtual tours. We do tours here every year. This is the first virtual one for us, so we're glad this seems to be working out so far. My name is Adrian Feltz. I work here at Signature Science in Charlottesville. Our company is headquartered in Austin, Texas, but we have a host of clients here in Charlottesville that we serve. We've got about 90 people here. We primarily hire scientists and engineers and IT professionals, software engineers. But what you're going to hear today are from two teams of our chemistry and biology groups. We do a lot of forensic science, as well as analytical chemistry. So we have Doctors Gerton and Gordon, who are going to be talking to you about forensic science. But first, we're going to start off with Dr. John Canino. He is one of our resident chemists, and he has a really cool job. He basically, well, you'll see. He's got some videos for you to watch, and you guys can also send questions to your teacher, so she'll get them to us and be able to get those back to you. But just also really quickly, the Charlottesville Business Innovation Council does the tech tour every year. And if you participate, you're also eligible to apply for a scholarship for when you go to college. So I encourage you to submit an application for that if you so desire. So I'll turn it over to Dr. Canino now, and he can talk to you a little bit about what our analytical chemistry group does here. Yes, ma'am.
I'm having a little bit of trouble with the the view. So I'm trying to figure that piece out. And it keeps not showing up on the big. So I'm going to ask those of you at home, can you see it?
You have to do full screen. No, you have to full screen that window down there.
This isn't the window.
Oh.
Yes. It was full screen. It's not sharing. Then I can see you. All right, and this is. Oh, let's close it. Full screen. All right, and so then it should be able to share. So students at home, I want to ask you, can you see the screen with?
We can see your background, your wallpaper.
Okay. All right. Here is. Then. Oh, Ryan, you can see your name. This is what I'm not understanding. We're not able to share this. This is not because it's Zoom. It's what?
I said because it's Zoom, so I don't think you can share the Zoom screen on Zoom.
Oh, that's. Actually, if. Let's see, what I'm going to do is. Into Spotlight for everyone. I'm going to pin to first screen. We're going to try this view overview.
So we are each dialed in from separate locations.
Yeah. What I'm going to have to do is make one of you the co-host, or make you the host, So then we can share, you can share from your screen. That's the only way I can think of to do it.
Okay. John and Stephanie, why don't you guys rename yourselves so we're different? So that we know who to make the host. And John will be first.
And then, John, what you'll do then is when you're done, you'll just make, I apologize. Brain is too full of too many different things right now. So which one should I make the host? Actually, Adrian.
Yeah, the one that just unmuted. Let's go with that.
There you go.
Yeah, Adrian, I just made you host. So now if you make, go up to like 3 little dots at the top and then make John host. I'm gonna start share. Here we go.
Okay, John, I just made you a host, so you should be able to share your screen.
Okay,
There.
I just shared presentation, should be visible.
So Ryan, somebody at home, can y'all see the screen now?
Yes, we can.
Excellent.
Yes.
All right, well, let's get started then. All right, so my name is John. I work in the training group here at Signature Science, Charlottesville location. We do training courses in chemical, biological, radiological, nuclear, and explosives for first responders and military units. So today, we're not going to cover all of that because that would take a long time. We're just going to talk about the part I do the most work in, which is the explosive threats. So before we talk about that, we should talk a little bit about the chemistry that's involved. And in any kind of burning or explosion or any kind of reaction like that, what you're getting is an oxidation and reduction reaction. An oxidation is something that is losing electrons, and the reduction half is gaining those electrons that are lost. And in this case, we're doing that to generate gases, and we're doing it to generate gases very quickly. So if you think about burning a log in the fireplace, that's just a regular burn. It's kind of slow, it takes a while. If you were to speed that same thing up and generate all the smoke and all the heat and all the light that you got off of that log and made it happen very fast, it'd be something called either a deflagration or a detonation, depending on whether it's going below or above the speed of sound. So these are just really, really fast combustions, really. And so that they all kind of behave the same in terms of what's happening. So this is important, particularly for first responders. So if you have a fire group in here, they may be looking at entering a building, particularly a building like the one I work in right now. It's full of chemicals. And if it's on fire, they're going to want to know what those chemicals are so they can check to see, are things going to start exploding when I go in there? If I spray water into a room, is that going to cause an unexpected reaction? What am I worried about? We also do work with the military where they're concerned about how do I make explosives and what does it look like? And also police, if they walk into a clandestine lab and they see a bunch of equipment, it's nice to know what kind of stuff is being made there, or at least be able to kind of have an idea of it so you know what dangers am I looking at right now. So chemical incompatibilities can cause violent reactions. You can have strong oxidizing fuels that mix together. You can also have explosives and acids. We have all these things here at Signature Science. And so you can see if the building catches fire, it'd be a real problem. So I'm going to start with a few videos of self-igniting mixtures. So these are the kind of things that you worry about if you're a firefighter, you're going into a scene that has a lot of chemical storage. These are the kind of things that cause you a problem. Is it playing for anybody else? No? Why is it not playing?
No, not yet.
What? Okay, that's fine. All right, that's fine. Just over here. Okay, cool. I would check your videos to see if they work. Just give me a minute. We've got the videos over here. I'm going to stop sharing that. I'm going to go over here. You're going to share.
When you share, there are two buttons at the bottom that would allow you to share sounds and share video.
Yeah, it's the video is embedded in PowerPoint. It should have played, but apparently something is causing it to not.
Okay.
So we're just gonna see about trying to play it without that. There we go. Okay. This is gonna be a little rougher, take a little longer, but that's okay. Here we go. Just share entire screen. Okay, so here is. And you're seeing Alex, my lovely assistant in there, doing the mixing for me while I'm recording this. So he's mixing solid and liquid there inside of a fume hood. Takes a minute. We'll see a reaction start. So some of these reactions are quite slow, and this is actually where the biggest danger is. When you mix two things that go slowly, it means you have a chance to, if you're trying to, say, fight a fire, walk down the hallway after two chemicals have mixed and not realize that the fire is going to start up behind you. So stuff like this is actually more dangerous than some of the stuff we'll see in a minute, where the reaction is more immediate. Something like this, you may think there's no problem, and then suddenly we'll have a problem later. And there we go. So in this reaction, you can see a lot of smoke, but not a lot of fire. So it got hot, generated a bunch of gas, but it was kind of slow. It's kind of slow to start. It didn't generate a whole lot of heat. It generated a lot of gas. So this is something more like a pyrotechnic that you might see on the 4th of July. Fireworks generate a lot of light. Sometimes they generate a lot of smoke, depending on what you're trying to do. And we compare that to something, let's see. Yeah, we'll go with this one next. Methanol. So something like this, very fast. It has a lot more energy to it. All right. So to compare all those to the last thing I've got here. And this is Alex running himself a little experiment, testing some compatibilities. So he's got 2 salts. He's going to mix them together, and then we're going to watch it to see if anything happens. So he adds his 2 vials together. Gives it a little shake. It's the salt, so you got to bang it a little bit to get it to mix up. All right, so he's got it mixed up. Then he doesn't really get any kind of heat or any kind of smoke, so he's going to add a second. This is a metal now. Metallic powder. And he's going to spend a bunch of time mixing it together, so we're just going to skip a little bit of this. Don't tell him I skipped all of his excellent mixing work. He's very thorough. Yep. All right, so again, this doesn't really do anything. We're not really getting any kind of reaction. So now he's going to go clean it up. He's going to add some water in there. And the water is actually what starts the reaction. So you can see if you're somebody who is going to walk into a building and spray a bunch of water around to put out a fire, something like that would be a real concern. So that's the kind of thing we do with like a fire department. Here we go. We'll see if some of these work. I don't have a backup video for some of these, so hopefully they'll play. But yeah, I was convinced. No. So here we have, I'm just going to talk about it. Here we have some tests we do, small scale safety testing or sensitivity testing. So this is for people who are maybe bomb squad members. And so we make explosives that are known to be made by bad guys, and we give them a little amount and they can test how sensitive it is. And that lets them get an idea of how dangerous they are. You do things like test it with impact, which you can see a couple of people doing there. You hit it with a hammer. Friction, you rub it on that cinder block you can see in the background there over here. Heat is really easy. You just hold a flame to it. Same thing for the flame. Heat and flame, really the same test. Just like take a torch, hold it up there, and see how it burns. And some of these things are interesting where, you have an explosive, but it doesn't actually burn. Actually, quite a number of them don't. Electrostatic discharge is something you test on a specialized instrument, but you're basically seeing, hey, if I'm real staticky, is it going to be dangerous? Am I going to be at risk? Do I need to worry about grounding myself? And then chemical compatibility is the last one where you test it to see, does it react with acids? Does it react with metals? Does it react with bases? That kind of stuff. Unfortunately, it doesn't look like we can play our videos at all if they're in PowerPoint, which is annoying. So I had some more videos of stuff. Oh, but that doesn't work either. So here, the last thing is, if I am a police officer and somebody says, hey, I see a bunch of smoke coming out of a building and I don't know what's going on, or you're called to a scene and you go in and you see a lab going on. We teach people things to look out for. So thermal runaway is something that happens when you leave a reaction untended or you do the reaction wrong. It can cause it to go wrong and cause fire or smoke or toxic gases. And so in this first picture here, we have a reaction that is releasing toxic gas. And so this reaction has gone slightly wrong. Somebody's done something they weren't supposed to, or they weren't paying attention, or maybe they left because they saw the police were coming. And it's now, this reaction is going, but it's now releasing toxic gas. And so we teach people what to look out for. What are the signs of like, hey, somebody was doing something, and now it's not the danger of what they were trying to do, but it's the danger because they stopped paying attention to it. And so now it's dangerous for a whole different reason. This next one, which unfortunately isn't going to play, is a video. It's a very similar situation. You start seeing this nasty brown gas evolve, but then actually the reactant, the material being made catches fire inside the vessel. And so those are the kind of things we teach people to look out for. I think with that, because I don't think our next videos are going to play, I will take any questions people have. I don't think I can even get to the next videos. Yeah, we do some work at a range here in Virginia. Fortunately, none of these work. I don't know why, but they don't. So does anyone have any? Yes.
Sorry, it's Adrian. And I'm having
No
some
problem.
echoes, but I was hoping you could give the students an idea of what types of skill sets are required for this type of job that you have, and maybe what you
Sure.
studied in school.
Yeah, so I studied analytical chemistry. I went to the University of Rhode Island, both for my undergraduate and graduate degree, but chemistry is really what you would want to study for this kind of thing. You could also do organic chemistry, which is more of a doing the chemistry of doing reactions. Analytical chemistry is more the study of what chemicals are and how they made up and identifying the unknown chemicals. But you could also do organic chemistry, which is more of a focus on trying to make new chemicals or find new ways to make chemicals people use every day. Either one of those is a good way to go to do this kind of thing. Yeah, so Alex, my lovely assistant there in one of the earlier videos, actually this guy right here, did 5 years in the Marine Corps. And then when he got out of that, he decided he was going to go study chemistry in college. And that's how he got started at Signature Science. So there's a couple of different routes you can get there. Did we have any other questions?
But feel free to, if you're writing them in chat, make sure that everybody can see them. I'm just curious, do you, when you're working with like police or things, do you ever do like run-throughs or practice with them like in real time?
Yes, that's actually a large part of what we do. We do quite a bit of building of fake labs. So we'll go somewhere off-site and rent some space, and we'll set up an entire fake laboratory for them to do a run, for people to do a run through, try and identify what the process is, practice their own safety guidelines and how to handle stuff, how to handle evidence, anything they want to practice. And we set it up all with fake chemicals, so we're not, at risk of setting somebody's space on fire. But it lets people go in and practice looking at a scene, identifying stuff, collecting any evidence they want to collect, and any of the other practices. So we do a lot of work with civil support teams, and every state has at least one civil support team, and they're a Army Reserve unit that is assigned to deal with anything basically the state's first responders are overwhelmed by. So if it's maybe it's some sort of a big chemical spill and the state just doesn't have enough local resources to deal with it, they'll call these guys in to help. A lot of them were called in to help with COVID. They did a lot of testing and diagnosing of people. They do all kinds of different stuff, but they also do a lot of responding to just like, hey, we ran into this chemical lab, this illegal chemical lab, and we have no idea what it is. So can your guys come by and take a look at it? And so they wear very heavy protection. You actually can see them in that first slide I had up. Go back over to it. That's actually them there. And that's in what they call level A protection, which is entirely self-contained. They've got their own air tank in there. That's what's underneath this big bulge that you see there. And then you can see they've got a mask on. So all of their equipment, all their kit, the radio, everything they've got is contained inside this protective suit. So when they get out, they can just peel that off and have deconned and gotten rid of. Because they don't know what they're walking into. So it could be fine. They may not need that at all, but they don't know when they get there. So they wear these really big, heavy protective suits until they can identify what's going on. And then they may choose to downgrade their PPE.
And then also, are y'all dealing with like setting up for like meth labs, drug labs that people might have?
Yeah, we've done some setups doing stuff like that. It's pretty easy to do a pretend lab setup. Doing a real one is harder, but doing it just to make it look right is actually pretty easy. It's mostly a lot of trips to Lowe's.
Well, I think somebody said that their spring break, that was a lot of trips to Home Depot, but but does anyone else have other questions? Because this is always what you guys ask me, or students ask me at the beginning of the year, are we gonna make anything blow up? Are we gonna be mixing chemicals together? And I'm like, that's next year, folks. So this is what we're talking about.
I asked the same question when I was in high school chemistry, and I was told no. So I had to stick with it for a while longer before they'd let me.
Now you get to blow stuff up, or safely. Well, folks, if you think of another question, or it's, you've got something in the back of your mind, but you can't quite put it in words yet, just save it, write it down and share it with me in email, or share it with me tomorrow in class. And then I will pass it on.
Okay. All right, can you hear me now? Hopefully without the feedback this time. All right, let me share my screen. Going on.
Steph's having some slight technical difficulties. She needed to switch a permission to allow Zoom to see her screen. She'll be with us in a second.
All right. If you saw some of the other things that Signature Science works on, they were.
Can you folks hear me now?
Oh, yeah, we can hear you.
Is this working better? I think you
Yeah.
can-
We can see, yeah, we can see Singing for Science, but we can't see your screen.
I can't hear anyone else. Can anybody hear me?
We can hear you.
I can hear you.
It's telling me that I can't share my screen still.
Miss Rigby, you might have to allow her to.
Yeah, I don't have that
I did
capability anymore. It has to be the host. So whoever is the host needs to go
I
down.
did that.
You can do that?
Yeah, I did.
Okay, Henry just did that, so you should be able to share your screen now.
I'll do it.
They're not moving, it's just fingers.
I can't tell.
I'm the one that's Signature Science, Stephanie.
Seriously?
Yeah. Okay, so I think I can share my screen now. There we go. That's much better. All right. So my name, as Adrian mentioned, my name is Stephanie. I'm also here with Aaron Gordon, who's another one of our DNA folks. We don't have any videos of anything exploding, but hopefully my videos do work. So So first, I wanted to show you a little of our lab, which actually we can do better over Zoom than we could do if you were here. I'll explain why in just a second.
Hi, I wanted to give you a tour of our DNA lab and talk a little about what we do here. But first, a couple of basics. You've probably learned about DNA in your science classes already, but the really interesting thing about human DNA as a scientist who works on DNA is that there's very little difference between the DNA in your cells and the DNA in your neighbors and your parents and everybody else on the planet. It's only about 5% difference between you and anybody else. So what we do here is look at that 5% and see what it can tell you about the person that it came from. Your 5% difference is what makes your hair color, your eye color, your height, a whole bunch of other things that are specific just to you. Now, we are not a crime lab like you may have seen on CSI. What we primarily do is research and development, R&D. So what we're trying to do is take the existing technologies and make them better for the people who do work in labs like that. So in this lab, we have a bunch of different technologies for examining DNA. And they all work in different ways. They can tell you different things with different starting types of DNA, and then give you different information at the end. One of the really neat ones is this little bitty sequencer called a mid-ion, little bitty tiny guy. This is another sequencer. This is called an iScan. There's yet another one over here, Nexi. They all work in very different ways. So if you're interested in that kind of technological development, where you get into the nitty-gritty of the hardware and software and how it works and how people use it, this is a great kind of job for you. This is what I really enjoy doing. I can take it apart, I can see how it works, I can put it back together and make it do something that maybe somebody didn't think it could do before. Or I can make it do the things that it already did, but better, easier, faster.
Okay, and then I have one more.
Hi, I wanted to give you a tour of our DNA lab.
That's the wrong video. Did it not skip to the next slide? Next slide. How do I, too far, is this the right one?
So the other question you might have.
You'd never believe that I work on computers all day long.
So the other question you might have is how you get this job. A lot of the people I work with have very, very different backgrounds. And I have to admit, a job that I never thought I would actually have. So I started out as a marine biologist. I wanted to go to sea and work on boats, but then I discovered that one of the other things that DNA does is change how your brain works, even down to the very tiniest animals that you don't think of as having a brain. I worked on jellyfish. But once you start looking at how DNA differs between different kinds of animals, and then between people within or different organisms within a species, that's when you start really diving into how does it work? How do I take it apart? How do these pathways go from here to here to here and make an eye in you or an eye in a jellyfish with the same DNA underneath? Some of my other coworkers, like Erin, have very different backgrounds from mine.
Yeah, so my background is more traditional biology. I majored in genetics and then went to school specifically for forensic science. So I knew that I wanted to work in a lab similar to this, whereas Stephanie probably kind of came to forensics through a much, much different pathway. So I think I have more maybe like traditional biology and, well, biology in the sense of forensic biology, whereas Stephanie has a lot of cool different experience doing all sorts of other stuff, which she can also tell you about.
So when you think about genetics and you think about working in a lab like this, it's important to remember that sometimes it's not how you get here, and sometimes the way you get here won't be the way you thought of initially.
So the other thing I wanted to show you today is a little bit of how we actually do that DNA analysis once we have it. So we do your sequencing. If you've ever watched CSI, you might see them holding up those bands against the little light box, all the little blue and white bands. So what does those bands actually mean, right? What we're looking at there is what are called STRs, which are short tandem repeats. They're groups of repeating bases. And at each STR site, each person has 2 copies, one from your mother and one from your father. And each person's combination of those 2 alleles is unique. If we look at all the STRs in your genome versus all the STRs in the genome of any of the other 7 billion people on the planet, they are exactly unique. We get them by using another instrument that you couldn't really see in that video that I showed before. Okay, this is a capillary electrophoresis instrument. And what it puts out is these DNA profiles that look like this. And it's measuring the fluorescent response of each of these different allele sites. So what I'm going to show you is how we compare them. So if you have 2 samples, say you have a piece of evidence that you collected at a crime scene, and then you have somebody you think might be responsible, you would run both of those DNA samples through that instrument I just showed you and see how these peaks compare. So do they have the same peaks here and here? So I thought I'd give you a little model case, right? So say one day the Stanley Cup comes up missing. Who took it? And we have all these potential people, all these potential team mascots who maybe ran away with the Stanley Cup. So you get the Stanley Cup, you swab it with a cotton ball, you extract that DNA, and you get this profile. It shows you these bunch of peaks. So someone with these STRs took that trophy, or at least touched it. But now you need to know who that might have been. So you go out, you take some samples from all the people who might have taken it, all the mascots. So up here on the top line, we have the swab from the Stanley Cup. Down here, we have person number 1. So do you think, I guess I can't really do polls through Zoom. So think about as you look at this, do you think that person 1 there matches that swab sample? And I'll tell you how we look at it. So one of the first things we look at is this part right here, okay? This X and Y. If you've learned in biology class about X and Y chromosomes, so, you know, most people who are girls have XX, most people who are boys have XY. So our sample off the Stanley Cup has an XY, probably a boy. This potential person down here only has an X, probably a girl. Probably not the person who took the Stanley Cup. Okay, how about our next person? So this person again, XY here on our sample from the Stanley Cup, XY in our potential sample, but then we have to look at these other loci, and that's what I mean when I say everybody's unique. So you might have the same X and Y here, but you're gonna have something different here or here. So in this sample, you can see this individual here that touched it has a 28 and a 31. That means they have 28 repeats on one chromosome and 31 repeats on the other chromosome. And this person here that we thought might have taken it has 30 repeats on one chromosome and 33 on the other. So we can tell that that's not the same person either. We're running a little long, so I'm going to skip straight to person number 4. And we can look again. So here we have an X and a Y. Now we have a 12 and a 13 here. 28 and a 31, they're all matching. 12 and 15, all matching again. 11, all matching. So they all matched up. And we can say that with certainty, out of the 7 billion people in the world, this is the person that must have touched that object, because they're the person that's going to have this unique combination. We already mentioned a little bit some of the things that you can do in this field, some of the ways you can get here. So you can be a DNA analyst, a forensic scientist, just like you've seen on CSI. You may have seen people getting 23andMe genetic testing. One of the other new careers in this is genetic counselor, where somebody gets a DNA test from a company and they need to know what it means, right? You don't have to be a genetic specialist. Can you call somebody who is? And then, of course, there's professors and teachers and writers and a whole bunch of other things. I see I'm getting things in the chat here. How do I get to those?
Yeah, it's just reminding folks that we have 1 to 12:05, but we're going to continue, like we talked about yesterday, we're continuing through, and then we'll go out, we have break at 12:25. So.
Does anybody have any questions?
I was like wondering how much DNA does it take to figure out? Because I'm thinking of all the people who could have touched the Stanley Cup or had a great out of it. How much DNA does it take to be able to figure out this?
So that's actually one of the types of R&D that we do here, is we work to make it take less and less DNA. When these technologies started, you needed a lot of DNA. People would use drops of blood, or they would use chunks of skin. If you've ever done a 23andMe DNA test, they use a lot of DNA, and they take a Q-tip and they run it around inside your mouth. It has lots and lots of DNA on it. We can now get DNA profiles down to just a couple of cells worth of DNA. So a very, very small amount. One of the other things we do, because you mentioned that a lot of people would have touched the Stanley Cup, one of the other things we do is try and figure out mixtures like that. So if more than one person has touched an object, how do you tell what parts of that DNA sample came from one person and what came from another? That takes a lot of math and statistics. So if you're really interested in math or you're interested in statistics, that's the kind of job where you have a lot of fun trying to split those apart. Anybody else? And if you do have any questions later, you can always send us an email and we will be happy to answer questions you might have later.
I really appreciate the slide and the information about being working with DNA. What are all the other options that you can have? And I think that's one thing that as you get into careers and doing different things, sometimes you realize it'll take me down a different path. It sounds like you started with marine biology, but then now you're working specifically with DNA. So it's really valuable.
Yeah, I think Signature Science employs, and I mean, a lot of forensic labs might as well employ a lot of different types of individuals. So it's not just maybe your traditional biologists, but yeah, people who are IT specialists, people who are, like Adrian said, engineers, or a lot of other types of jobs as well, or backgrounds, I guess, as well, can all come together and work in the same place.
Yeah, everyone, if you have questions, please let me know, and I will pass the questions on.
Well, we appreciate the opportunity to talk to everybody. Is that all you need from us, Ms. Rigby?
Yeah, that's fine. What I'll do is hopefully it recorded.
Hi,
And
mozzarella.
I got just yours. So yeah, lunch just arrived. So I will let you know. And you guys, thank you very much for everything. And technology is always learning every day. And then awesome. Thank you very much. But then and I will see you guys tomorrow afternoon. Hopefully the recording works. So we'll see how that turned out.
Okay, that sounds great. I'll look for it and we'll see you tomorrow.
Sounds good.
Great, thank you.
You're welcome. Thanks.
Bye.
Bye-bye. So everyone here, what I want you guys to do is we'll have lunch. At 12:40, if you guys can pop back on just for about, I'm close now. You know what, here's the scoop. Do you want me to go ahead and tell you what you're doing? And then you don't have to come back after lunch? But I'm not sure who else here. So what would y'all like?